Welcome geniuses, I'm Genie, your best buddy for A-levels. In this channel, we'll bring you to explore the secret formula behind success.
Okay, today I'm going to cover the topic on AS Topic 1, Calibration of Eyepiece Graticule. So, when you are supposed to calculate the actual cell size, what you need here is, you need a stage micrometer where it looks like a slide but with a small ruler with a known value embedded on the slide itself. And second thing that you need will be your eyepiece graticule, which is a scale that can be fitted into your eyepiece lens. But usually you wouldn't get to see the eyepiece graticule because it's already been fitted into your eyepiece for you. So now we just go into the setup. So in your light microscope, you will see that your eyepiece already has a lens in it that magnifies at 10 times. So inside the field of view, this is what you get to see. So when you zoom in, that's where you need to observe how many divisions are there in your eyepiece graticule. So take a closer look. By looking at the number, you would think that it only has 10 divisions. But look into the details, you actually see that between 0 and 1, there are actually 10 smaller divisions. So in total, your whole eyepiece graticule is actually consisting of 100 divisions. So consider every small division as one division. Then what you have now here is, once I identify the number of division, so usually you will put your slide with the specimen under the light microscope on the stage. And for example, you are carrying out an experiment on cheek cells. So you will see many cheek cells within your field of view. So you're expected to calculate the actual cell size of your cheek cell. So most likely, you identify one cheek cells that you want to find out the size. So you can align the eyepiece graticule in such a way that it's like just nice. The number is like a whole number, or it's between the lines. But if not, it's okay. You just try your best to identify the number of division that matches the cell size. So like for example, in this case, the cell size is about, between here to here, you have 10 division, then 11, 12, 13, 14, about 14 divisions of eyepiece graticule. But this doesn't give you any value, as it's just the number of division. So to give this number of divisions a value, that's where we need to calibrate the eyepiece. Using what? Using a stage micrometer. So every stage micrometer might not be the same. So you have to look at the indication where they might tell you that this particular ruler or this particular micrometer is actually made out of what size, and therefore each division represents what. So in this case, the 1 mm, that means it shows that this ruler is actually 1 mm in length, divided into 100 parts. So the 100 parts is like 100 division. So if you magnify, you can actually see that this is the smallest division. So we need to find out what is the value of the smallest division on the stage micrometer. So if you do a calculation, 1 mm divided by 100, that's where you get 0.01 mm for each division of on stage micrometer. So now, moving on, we're going to use this stage micrometer to give a value to your eyepiece graticule, and that is known as calibration. So we're going to place the stage micrometer on the microscope stage, and under the field of view, you will see that The stage micrometer, you can see the scale of the stage micrometer and the eyepiece graticule together, but then it might not be aligned, so you have to adjust them in such a way that they are both aligned to each other. And better still, if both of the lines can be a match. Where you can see this line of the stage micrometer is matching the line of your eyepiece graticule. So if you can get a match, okay, it might not be necessarily the whole eyepiece graticule. As long as you can calibrate a portion, that will be good as well, because it's all evenly spaced. So any portion that you can calibrate, it will represent the whole eyepiece graticule. Then another thing that to take note is, if you view your cheek cell under 40 times magnification just now, you make sure that your calibration is also done under 40 times. So how can you identify which one is stage micrometer and which one is the eyepiece graticule? The scale that doesn't change under the light microscope will be your eyepiece graticule, because it's fitted in your eyepiece, so it doesn't change regardless what magnification that you change using the objective lens. So for this, the larger scale will be known as your stage micrometer. Size will actually change according to your magnification, according to your objective lens. So in this case, if you have viewed your cheek cells under 40 times magnification of your objective lens, you make sure that this calibration is also done under 40 times. So now, let's calibrate the eyepiece using the stage micrometer. So you can observe that here, you have 10 divisions of stage micrometer. That is matching with, okay, how many divisions of eyepiece graticule? From here to here, we have a total of 10, 20, 30, 40. So I have 40 divisions of eyepiece graticule. So how am I going to give or calculate the value for each eyepiece graticule? So previously we have already calculated that each division on the stage micrometer is actually 0.01 mm. So now for 10 division in length, so what is the length of this whole 10 division? It will be 10 times 0.01 mm. So knowing the length of this stage micrometer that matches the eyepiece graticule, I can actually calibrate the eyepiece graticule. So we're going to start the calculation now. So to find out the value of each eyepiece graticule division, it will be equivalent to the length of the stage micrometer that is matching. So I have 10 division times the value of each division and divided by how many divisions of eyepiece that is matching. So if you carry out the calculation, you realize that the answer is 0.0025 mm. But like I said, since it's under the light microscope and we're going to view microscopic specimen, so the unit has to be converted to micrometer, where you have to times 1000. Therefore, you get 2.5 micrometer. So each eyepiece graticule division is worth 2.5 micrometer. So I go back and show you. So after we calibrated, it means that each division on the eyepiece graticule here is 2.5 micrometer. So after you've identified this, now we're going to calculate the actual cell size. So for the actual size for the cheek cell, So can you remember just now how many divisions is our cheek cells? It's actually 14 eyepiece graticule division. So now we know that each division is 2.5. So I just times 2.5 micrometer. That's where I'll get 35 micrometer. This will be the actual cell size for the cheek cells. So if you want to know like a general formula, the general formula will be to identify the actual cell size. It will be, you need to identify the value of one division on stage micrometer times the number of division on the stage micrometer that is matching with the eyepiece divided by the number of division of eyepiece graticule unit that is matching times the number of division occupied by your cell. That will be the formula. If you feel that sometimes you are very confused, you can remember this particular formula and try to derive the answer from there. Okay?
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with one or two past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.